Here are the most unsettling crossbreeding experiments from hell. Have you heard of any disturbing experiments? Let us know below. Number 5. Cyber beetles. While this may sound like a science fiction future, we are already living through it. In 2009, DARPA, the Defense Advanced Research Projects Agency of the US military, revealed that they had already successfully turned rhinoceros beetles into computer chip-controlled biological UAVs. The chips were implanted into the brains and had a receiver and transmitter to communicate to a computer, which would tell the beetle to fly up, down, left, right, forwards, or backwards. At the time, the project was in its infancy and was limited in its use. However, it has since been 14 years, and scientific advancements since the late 2000s have been massive, especially in the area of computers. It's believed the same process can be done to rats, which effectively opens the door for remote-controlled biological weapons or spies, which could be a very real aspect of future war. Back then, the size of chips and transponders meant that the beetles couldn't carry much more than the bare minimum and still be able to fly. It is now possible that beetles could have cameras and other surveillance equipment implanted and are being used as biological spies. Conversely, an army could equip a swarm of beetles with biological or chemical agents and be deployed in a buzzing swarm against opposing forces. The possibilities are truly disturbing and endless. From a spy system based solely on insects, which can spy in both foreign and domestic targets, to a swarm of weaponized rhinoceros beetles. Number 4. Pig-grown limbs. The latest advancements in stem cell research have allowed scientists to stop using human fetal tissue or test tube-grown embryos for stem cells. It's been discovered that the lining of pig bladders and intestines actually contain stem cells. This biological matter can be isolated, dried, and ground up to create something called an extracellular matrix. This stem cell powder has actually been used to encourage digit reattachments in cases where people have lost their fingers. There's a hope that this new source of stem cells could be used eventually to fully regenerate damaged or missing limbs and organs. Some believe the next direction would be to start using these pig stem cells along with human stem cells to allow organ starters to be implanted in pigs and allow for organs to be grown for transplant that are genetically identical to the host and thus won't be rejected. However, there are concerns surrounding the ethics of using live animals as hosts to grow limbs and organs in, and the potential ecological or ethical issues that may arise from profit-driven human-pig limb farming. Number 3. Yersinia pestis. Known better as the Black Death or the bubonic plague, Yersinia pestis has been responsible for some of the worst plagues in human history and billions of deaths. The bacteria can be acquired either through breathing in the bacterium, which is called pneumonic plague, or through flea bites called bubonic plague. Scientists have brought Yersinia pestis back numerous times to study. However, new genetic engineering technology could open up the door to a new and terrifying plague. There are 3 main factors that determines the effect of biological weapons: environmental persistence, infectivity, and lethality. Unaltered Yersinia pestis can stay in infected soil for up to 24 days in the right conditions. It's also highly transmittable with an infection rate of 1.9 and is already known clearly to be highly lethal. Biological weapons like Yersinia pestis have been used for centuries, as recently as World War II. However, they have always been more dangerous to wield due to their infectious and unstable nature. But with modern gene editing, it is possible to edit Yersinia pestis or any other biological threat into a multitude of weapons. A fast-acting, highly lethal, and infectious disease with very poor environmental persistence could be used to clear the path of advancing troops. A highly persistent infective strain with treatable but long-lasting symptoms could be used to protect the flanks. Or in the worst-case scenario, a highly persistent, antibiotic-shielded, highly infectious strain with a long death period could be used to target enemy population centers. These possibilities are truly more terrifying than the worst chemical or nuclear threats, as they have the potential to commit accidental autogenocide. A perfectly crafted bioweapon could be too effective and wipe out all of humanity. Number 2. The Belgian blue cow. These cows were bred in Belgium for a gene which causes double muscling. Double muscling, or hyperplasia, is caused by a gene which increases the number of muscle fibers instead of increasing the size of muscle fibers, which would be hypertrophy. This allows Belgian blue cattle to convert feed into lean muscle more quickly and efficiently than any other breed of cattle. Belgian blues were first bred in the 19th century and are now bred in over 20 countries. They're prized for their high muscle to fat ratio and fine muscle texture. But Belgian blues run into the same issues as most thoroughbred breeds, suffering a collection of genetic defects which make life very difficult for them. Double-muscled cows frequently have dystocia, a genetic condition which narrows the pelvic opening significantly. This means that essentially every Belgian Blue to have ever been born had to be C-sectioned. As well, the bulls have significantly lower fertility, which also causes issues in mating, though the dystocia tends to have a much more significant impact. Belgian Blues that live beyond their factory lives suffer serious joint and tendon issues as a result of the added mass, and would eventually collapse under their own weight. Not a nice way to go. Number 1. Persian cats. Persian cats are some of the most inbred and genetically diseased animals in the world, all thanks to human crossbreeding. They have shorter lifespans than other cat breeds, but that is only the beginning of their issues. Persian cats are genetically predisposed to be at higher risk of suffering coloboma, where the muscles of the eye don't fully attach, leaving holes, cataracts which can cause blindness, cryptorchidism which is both painful and causes sterility, urinary tract disease, polycystic liver disease, lupus, diaphragmatic hernias, and so much more. Persian cats are incredibly inbred, specifically for many of their more distinguishing deformities, such as their prominent underbite, which results in difficulties eating, and like pugs and bulldogs, causes Persian cats to easily run out of breath and suffer numerous respiratory issues. Even more disturbing, Persian cats are at a high risk of developing entropion, a highly painful condition in which the eyelids invert and scrape the eyes, potentially leading to blindness. The simple fact is that decades upon decades of selective breeding and inbreeding have caused Persian cats to be deeply afflicted with dozens of truly horrendous genetic illnesses and conditions.